Welcome back, friends. This is the third video of Environmental Mass Spectrometry series. In this video, we will learn about what mass spec can do briefly. So let's get started. In the second video, we learned that mass spec is a method for measuring the mass of an atom, or a molecule of the substance, so-called analyte. What mass spec can do is just measuring the mass of an ion, but by measuring the mass of ions, we can conduct so many things, like quantification, structural elucidation, retrospective analysis, microbial screening, analyte location imaging, mRNA-DNA vaccine analysis, and so on. Let's see each one by one. Surface water, effluent, air, food, blood, breast milk, and doping test samples. There are many kinds of samples to be quantified by mass spec. These quantifications must be accurate, precise, sensitive, and selective. Mass spec has the ability to determine an analyte concentration with all the features. Please note that accuracy is different from precision. Their sensory representations were illustrated on the upper left side. Accuracy refers to the closeness of a measured value to the true value. Precision refers to the closeness of multiple measurements to each other. Accuracy may be controllable by calibration. To calibrate your mass spectrometer, you usually have to prepare a set of authentic standard samples, containing a known amount of the analyte of interest. Mass spec response, such as abundance or peak area for each sample, should be recorded to establish a relationship between the response and the analyte amount. The obtained relationship is then used to transform the response observed on test samples into the amount of analyte present. This is the basic concept of a calibration curve, illustrated on the right side. Several calibration curves for several analytes can be obtained simultaneously, because mass spec is highly selective for different analytes. This unique feature enables us to quantify many analytes at one injection to a mass spectrometer. This is so-called simultaneous quantification. During sample preparation like adsorption, desorption processes, some analyte molecules may be lost in some cases. Mass spec can compensate for the losses by using stable isotope-labeled chemicals, so-called surrogates. For instance, the chemical structure of caffeine was drawn on the right side. Left is native caffeine, and right is carbon-13 labeled surrogate. If the detected surrogate amount was smaller than the added surrogate amount, the surrogate was lost through the sample preparation processes. The lost percentage, that is, the recovery of the surrogate, must be identical to that of the native caffeine, because the chemical properties of both the native caffeine and its surrogate are almost the same, despite their different masses. The compensation by using surrogates is a unique advantage of mass spec. The next what mass spec can do is structural elucidation. Structural elucidation of the environmental unknown is a very powerful technique. For example, Tian et al. Identified a highly toxic transformation product from the globally ubiquitous tire antioxidant, 6-PPD, by using mass spec and NMR. This is a good example of structural elucidation. Mass spec structural elucidation is adaptable to unknown disinfection byproducts, unknown photoproducts, unknown toxicants, illegal dumping, illegal food additives, illegal drugs, toxicants unintentionally produced during recycling, drug metabolites, and so many targets. This is our structural elucidation manner for environmental toxicants. First, a comprehensive analysis, so-called non-targeted analysis, is going to be performed on a set of samples, containing various amounts of unknown toxicants. Second, computational analysis, like multivariate analysis or differential analysis, is going to be conducted, to explore causative toxicants, among numerous detected compounds. Third, molecular formula determination is going to be performed, on the explored causative toxicant. Then, structural elucidation of it is going to be performed with multistage mass spectrometry. This elucidated structure is going to be confirmed, with quantum chemical calculations of fragmentation reactions. And finally, the explored causative toxicant is going to be identified by co-chromatography. In this video, only the first and the second steps are explained briefly. The first step is a comprehensive analysis of samples, containing various unknown toxicants. Comprehensive analysis is also called non-targeted analysis. A standard analytical method for quantification is a targeted analysis. Usually, environmental samples contain many contaminants, A, B, C, etc., but targeted analyses detect only the focused contaminant. On the contrary, a non-targeted analysis is inherently capable of detecting all the ionized analytes within the scan range. In order to explore unknown toxicants, a non-targeted analysis is needed. By performing a non-targeted analysis, numerous compounds must be detected in one sample. We must explore the causative toxicant from among numerous detected compounds, by using relationships between the sample's toxicities and mass spec responses of each compound. For example, now we got 7 samples, posing different toxicity strengths, as shown on the upper side. Their toxicity strengths were described in the toxicity unit. Non-target analyses were conducted for each sample, resulting in the detection of compound A, B, C, and Z. Peak areas of each compound were of course different, like the figure on the lower slide. For example, the peak areas of A, orange-colored closed circles, were almost flat against the sample's toxicities. On the contrary, the peak areas of D, light blue-colored closed circles, almost correspond to the sample's toxicities. This correspondence implies that compound D is a major causative compound. In the actual procedure, we will perform multivariate analysis to explore causative compounds. Besides quantification and structural elucidation, mass spec can do so many things. For instance, mass spec can analyze an emerging toxicant retrospectively, by using the data acquired 10 years ago. Perform a quick screening of a major microbial in the sample, by laser desorption ionization without digestion. Visualize chemical distributions in tissues or flocks, by laser desorption ionization without digestion. Characterize the glycosylation patterns of the SARS-CoV-2 spike protein, in order to guide vaccine and immunotherapy development, and so on. Mass spec can go beyond the scientific or technological horizon. That's all. Thank you for watching.